In this video, I will share my journey in developing a material that enables the detection and differentiation of amine gases using a smartphone. To achieve this goal, I synthesized and tested various BODIPY derivatives with bromine atoms at the alpha position. Among these structures, the compound I named TolBodipy yielded the best results. First, let me explain how I synthesized this compound. To obtain the target compound, I first needed to synthesize the pyromethane. For this, I reacted pyrrole and paratolualdehyde in the presence of an acid. Theoretically, the synthesized pyromethane should be a colorless substance. However, due to its unstable nature, it rapidly forms byproducts, giving it an ash-like color. In solution, it appears colorless as expected. In the next step, I converted dipyromethane into the BODIPY core. To do this, I first oxidized with DDQ. Then I added triethylamine to remove the proton from the pyrrole ring. In the final step, I added boron trifluoride, which leads to BODIPY structure. The synthesized BODIPY compound is orange colored. However, compounds with strong light absorption typically appear darker in solid form. When dissolved in acetone, it exhibits an intense orange color. In the final stage of the total synthesis, I introduced bromine atoms at the alpha and beta positions of the BODIPY core. This yielded the TolBodipy compound. In its powdered form, it appears as a bright, intense black solid. After extensive purification, the final TolBodipy has a deep red color. In solvents, it exhibits a vibrant pinkish red hue. Under UV light, its solution emits extremely strong fluorescence. Now I will test TolBodipy selectivity for amines in solution. I prepare a solution of the compound in acetonitrile and distribute it into vials. Then I add different amines to each vial: ammonia, propylamine, pentylamine, diethylamine, piperidine, morpholine, and triethylamine. The color changes are quite distinct. Moreover, most amines cause a unique color change. Here are the vials under UV light. After waiting for half an hour, the color changes are complete. No organic substance other than amines causes a color change. So, what's happening chemically? The bromine atoms at the alpha position of the BODIPY core react with amines. For example, with a primary amine, a substitution reaction occurs, forming compounds that appear orange in visible light and under UV light. Secondary amines also undergo substitution reactions. However, due to the presence of two substituents, their oxochromic effects differ. As a result, the formed compounds appear blue-violet in visible light and range from purple to pink under UV light. Alpha halogenated BODIPYs react entirely differently with tertiary amines, forming a zwitter ionic structure where the BODIPY core is somewhat perturbed. These compounds appear yellow in visible light and emit a strong green fluorescence under UV light. Now I will transform TolBodipy into a material capable of distinguishing amine vapors. For this, I dissolve TolBodipy in acetone. I immerse thin layer chromatography plates into this solution, allowing TolBodipy to adsorb onto the silica surface. Now I will expose the plates to different amine vapors. For primary amines, I use ammonia and propylamine. For secondary amines, I use diethylamine, piperidine, and morpholine. The clearest differentiation among the same class of amines is observed with secondary amines. That is, variations such as cyclic structures and heteroatoms in oxochromes influence photophysical properties. For tertiary amines, I add triethylamine. Here are the final colors of the plates. The leftmost plate is the reference that was not exposed to amine vapors. Here are the plates under UV light. The plates with slight color differences in visible light become more distinct under UV light. But there is an important point. Amines with very similar structures can cause similar color changes. So, how can we distinguish these subtle differences that the human eye cannot perceive? This is where digital image analysis comes in. I converted the color changes caused by each amine into numerical data using red, green and blue or hue channels. Then I coded a program to process this data. With this method, a smartphone alone can determine whether a substance is an amine and, if so, identify the specific amine without the need for any additional devices. Currently, dozens of different dyes are used in smartphone-supported analysis. However, the method I developed achieves powerful differentiation using just a single dye. I published this study in Spectrochimica Acta Part A, Molecular and Biomolecular Spectroscopy. For those interested in details, I have included the link to the paper in the description.